Good morning, everyone, and welcome. I'm delighted to have you here today. We will be discussing range criteria in analytical method validation. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the course of this presentation, we'll explore the key aspects of establishing and validating range criteria, understanding their importance in analytical methods. We'll cover the fundamental principles, practical considerations, and regulatory expectations. I look forward to sharing insights that will help you in your work. Let's get started. Now, let's delve into what analytical method validation really ensures. Essentially, it confirms that a method is reliable and suitable for its intended purpose. When we talk about the key validation parameters outlined in USB 1225, we're covering a comprehensive set of criteria. These include accuracy, which tells us how close our measurements are to the true value. Precision, on the other hand, reflects the consistency of our results when repeated. Specificity ensures that our method can accurately measure the analyte in the presence of other components. Linearity shows that our method responds proportionally across a range of concentrations. The range indicates the interval over which the method is reliable. LOD, or limit of detection, is the smallest amount we can detect, while LOQ, or limit of quantitation, is the smallest amount we can measure with confidence. Robustness tests the method's resilience under small variations. All these parameters together help us achieve accuracy, precision, and specificity, making our method not just accurate and precise, but also specific, linear, reliable, and robust. These qualities are fundamental to ensuring the data we generate is trustworthy and fit for decision-making. They form the backbone of quality assurance in analytical testing, tying back to the overall goal of producing consistent, reliable results in our work. Let's talk about the definition of range, or USB, in the context of analytical procedures. The range is essentially the interval between the upper and lower levels of an analyte that we can reliably measure. When I say reliably, I mean that within this interval, our method demonstrates suitable precision, accuracy, and linearity. In simpler terms, it's the concentration window where our method works consistently and dependably. Now, it's important to understand that the range isn't just a random span. It's established based on how well the method performs in terms of linearity, how the response relates to concentration, accuracy, how close our measurements are to the true value, and precision, how consistent our results are when repeated. If these parameters are acceptable within a certain interval, then that interval becomes our validated range. So, in essence, the range is a key parameter because it defines the limits within which our analytical method provides trustworthy and reproducible results, which is fundamental for quality control and regulatory compliance. This connection between the range and other key parameters ensures that our method is both reliable and scientifically sound across the specified concentration span. Now, let's take a look at the typical range for different analytical methods. Starting with the assay method, we generally expect the results to fall between 80% and 120% of the label claim. This range ensures that the actual content is close enough to what we claim on the label, maintaining product consistency and quality. Moving on to content uniformity, the acceptable range broadens slightly from 70% up to 130%. This wider window accounts for small variations that can occur during manufacturing, but still ensures the product remains effective and safe for consumers. Finally, for impurity methods, the focus is on sensitivity and safety. We typically measure impurities from the limit of quantification, or LOQ, up to 150% of the specification limit. This approach aligns with general USB practices and regulatory expectations, helping us catch any impurity levels that could be problematic. Overall, these ranges are essential to ensure our methods are reliable, and our product quality remains consistent across different testing scenarios. Now, let's take a closer look at what we mean by accuracy within range. Here, we're using some example data to illustrate this concept. Suppose our assay range is centered around a target concentration of 100 micrograms per milliliter. The range we select around this target is crucial because it helps us determine how reliable our measurements are. When we talk about accuracy within this range, we're essentially asking, how close are our measured values to the true target value? If our results consistently fall within this specified range, it indicates that our assay is accurate and reliable for that concentration. This is important because, in real-world testing, we need to ensure our measurements stay within an acceptable margin of error. Consistency within this range means confidence in our data, which is vital for quality control and validating our methods. So, in summary, understanding how our data fits within this selected range helps us verify that our assay produces precise and trustworthy results around our target concentration. It's a key aspect of ensuring overall data quality and method validity. Today, I want to focus on the idea of precision within range. This concept is really about understanding that in many situations, achieving perfect accuracy isn't always necessary or even practical. Instead, what matters most is staying within an acceptable range of values or outcomes. Think of it like setting a target in a game. You're not aiming for perfection, but for consistency and reliability. As long as your measurements or results stay within that predefined range, you're effectively meeting your goals. This approach is crucial because it helps us balance effort and precision. Sometimes, pushing for absolute accuracy can be time-consuming or costly, while staying within the right range offers a more efficient and still effective solution. Ultimately, embracing precision within range allows us to maintain quality and accuracy without overextending ourselves, which aligns perfectly with our overall theme of smart, practical decision-making in our processes. Let's take a closer look at the concept of linearity within the range. When we talk about linearity data, we're essentially examining how well our measurements or responses follow a straight-line relationship within a specific interval. This is important because it tells us if our sensor or instrument provides consistent and predictable results over that range. The key point here is that linearity is not just about the entire measurement span, but specifically about the section where the device performs reliably. If the data points align closely along a straight line, it indicates good linearity, which means our measurements are accurate and reproducible within that range. Maintaining linearity is crucial because it directly impacts the precision of our measurements. When the data is linear, calibration becomes simpler, and we can confidently interpret the results without needing complex corrections. So, in the context of our overall theme, understanding and verifying linearity within the specified range ensures that our system or method is dependable for precise measurements, reinforcing the integrity of our entire process. Let's take a closer look at this example of an impurity method range. First, notice that the specification limit is set at 0.2%, and the LOQ, or limit of quantification, is at 0.02%. When selecting our range, we chose from 0.02% up to 0.3%, which is 150% of the limit. This broader range helps ensure our method is robust across relevant concentrations. Our validation process covers several key aspects. We test precision specifically at the LOQ to confirm consistency at the lowest detectable level. We assess accuracy at 3 points: 50%, 100%, and 150% of the limit to verify our method's correctness across the range. Additionally, we evaluate linearity across the full selected range to ensure proportional response. Now, establishing the validated range depends on meeting certain criteria. Accuracy must be acceptable, which we define as within 98 to 102%. Precision should be precise, with a %RSD or relative standard deviation of 2% or less. And linearity needs to be excellent, with a correlation coefficient, R, of at least 0.999. Putting it all together, the validated range for this method is from 80% to 120% of the specification limit. This means our method performs reliably within this window, giving us confidence in its accuracy, precision, and linearity across the critical concentration levels. Let's take a look at practical calculation of impurity range. When we're talking about impurities in a material, understanding how far they can travel is crucial. This helps us predict the impact on the material's properties and performance. The key point here is that impurity range depends on several factors, such as the type of impurity, the temperature, and the material itself. By calculating this range accurately, we can better control and optimize the material's behavior. It's also important to connect this to our overall goal of ensuring material purity and reliability. So, in essence, these calculations give us a practical way to assess impurity spread and make informed decisions in our processes. Let's talk about acceptance criteria. When we say the range is acceptable, it means that our results fall within certain predefined limits. For example, in an assay, we usually look for accuracy within about 98 to 102%. This gives us confidence that our method is performing reliably. We also check the percent RSD, or relative standard deviation, to ensure our results are consistent and precise. If the percent RSD is within the limits set in our SOP, or standard operating procedure, that's a good sign of reproducibility. Another key point is the correlation coefficient. It needs to meet the established criteria, indicating a strong linear relationship, which supports the method's validity. We also look for no significant deviations across different levels or concentrations, ensuring our method is stable and reliable throughout the entire range. All these criteria, accuracy, precision, correlation, and stability, are clearly defined in our SOP, which helps us maintain consistency and quality in our results. So, in essence, these acceptance criteria are our benchmarks for confirming that our analytical method is performing correctly and producing trustworthy data. Let's take a moment to understand the difference between linearity and range. When we talk about linearity, we're referring to how well a system's output is directly proportional to its input across a certain range. Essentially, if the relationship is linear, doubling the input will double the output, tripling the input triples the output, and so on. This concept is crucial because it tells us about the predictability and accuracy of measurements or responses within that range. On the other hand, the range refers to the span of input values over which the system maintains this linear relationship. So, even if a system is linear within a certain range, outside of that, it might not behave predictably or proportionally. Recognizing the difference helps us understand the limitations of measurement systems and ensures we're working within their effective operating bounds. Connecting this to the overall theme, understanding both concepts allows us to select appropriate sensors and measurement tools, ensuring data accuracy and system reliability. It's fundamental to engineering and scientific measurements because it helps us interpret results correctly and make informed decisions based on those measurements. So, in conclusion, the range essentially defines the working limits of an analytical method. It's the span within which the method can produce reliable and accurate results. This range is established through key parameters like accuracy, precision, and linearity, which ensure the method performs consistently. Typically, for assays, this range is considered to be between 80 and 120% of the expected value. When it comes to impurities, the range can extend from the limit of quantification, or LOQ, up to about 150%. Having a well-defined range is crucial for reliable pharmaceutical analysis because it ensures that the results are trustworthy across the necessary concentration levels. And of course, this concept is not just best practice. It's actually required under the USB 1225 guidelines. So, understanding and establishing the appropriate range is fundamental for maintaining quality and compliance in pharmaceutical testing. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention today. Your engagement is truly appreciated, and we look forward to supporting you moving forward.